Thank you. So thank you everyone for coming to this month's Nuance Tech Talk. Today I'm going to be presenting on behalf of BioCryo, and my name is Chris. I'm a postdoctoral research associate in both BioCryo and in EPIC-FIB. And today I'm going to be talking to you about common cell and tissue embedding protocols at BioCryo, in particular high pressure freezing, freeze substitution, and chemical fixation. Before we really get off to the races, I just want to note that BioCryo has a new RRID. So when you're publishing papers that have used BioCryo facilities or assistant, it is really helpful for us if you include this acknowledgement text here. This text is also available on our Nuance website under the publication acknowledgements, so you don't have to scramble and get a screenshot or copy this down now. There are RRIDs for all of the various facilities at at Nuance now. So please do use these when you're publishing papers that have used our facilities. It helps us a lot. Thank you. So you have a biological or soft matter sample that you want to characterize. However, because it's hydrated, you can't really put it into an electron microscope or other vacuum system very easily without contaminating the vacuum system and damaging your sample. What can you do? So BioCryo is here to help with exactly that problem. So let's start right at the beginning, because that is the very best place to start, with three really important questions for picking the technique that will be best for you. The first question is, how big is your sample and how big is your feature of interest? That will drive a lot of deciding which technique will work best for your sample. It's not the definitive answer. The other two questions are important for guiding this a little bit more, but it is probably the biggest one. Second, will your sample have inherent contrast or does it need to be stained? That will help us really tailor protocols for your sample in particular and what you want to see. And third, what is your sample sensitive to? Can it be dehydrated easily? Is it going to be very prone to osmotic effects? Are there things that you want to image in your sample that are extremely pH sensitive or polyvalent ion sensitive? Can your sample be sheared? All of these questions help us really pick which technique and protocol will be best for you. So if your sample is very small and you want very high resolution feature, or you want to see very high resolution features, plunge freezing is probably going to be one of the best techniques for your sample. This is limited to very small samples, so samples on the order of less than a micron or so. But you can get very high resolution reconstructions of nanoparticles and those sorts of structures with plunge freezing. On the opposite end, chemical fixation allows us to look at very large tissues and samples. It is an entirely room temperature technique, and it does have the highest chance of introducing a lot of potential artifacts. But in terms of just the total amount of material you can embed and start to look at, it is by far the winner. It also has the ability to work with the highest diversity of samples. So whether your sample is in aqueous phase or organic phase or dehydrated, we can really apply chemical fixation to your sample and give you something you can put in an electron microscope and start characterizing. However, if you need something that combines both of those worlds, high pressure freezing freeze substitution is really going to be your friend. It leverages all of the strengths of plunge freezing and the strengths of chemical fixation to really give you both high resolution and large tissues and structures. It's not quite as large of structures as chemical fixation, and you can't get quite as high of resolution as with plunge freezing, but you can get very, very good, very clean electron microscopy images with high pressure freezing freeze substitution. So this is a slightly overwhelming slide. Fortunately, we can ignore most of it for this talk, as we really just want to talk about these sample preparation workflows. And this is really where you will start your journey through the BioCryo workflow space with one of these protocols. Once you have your sample prepared by either plunge freezing, high pressure freezing, freeze sub, or chemical fixation, resin embedding, you can really then take your sample to almost any of the other sample cores in Nuance and start to do your characterization, or take your samples to other facilities as well. For example, I've personally taken cryo samples to the advanced photon source to do cryogenic wide-angle X-ray scattering after I did high pressure freezing here at BioCryo. So you really can take your sample anywhere else and work with them once you've gotten your sample embedded or prepared. So to really go into the details of each of these techniques, let's start with plunge freezing. So for plunge freezing, you are limited to aqueous samples only. You can't really work with samples that are in an organic phase. Additionally, you are limited to fairly low concentrations of samples. You want to be in the 0.1 to 10 weight% range. That's about 1 to 100 mg per mil. This is because to do plunge freezing cryo-TM really well, you want to have fairly uniform, well-dispersed samples only. If you have a sample that is highly prone to aggregation or is highly concentrated, there's a pretty good likelihood you won't be able to find an individual one of your nanoparticles or molecules or materials, fibers, whatever they happen to be, to image. If your sample is too dilute, it is very easy to spend an entire afternoon searching through a grid for any of the sample and find that your grid is completely sample free. So that's why we really want to be in this 0.1 to 10 weight percent range. Um, Alternatively, if you are working with bacterial samples, it is possible to do plunge freezing of bacteria monolayers. Bacteria are small enough you can actually plunge freeze them and get good structural preservation that way. And then you can prepare them in a variety of techniques to look at them with electron microscopy. In terms of our length scales, the samples that we can use for plunge freezing have to be less than about a micron in thickness, but you can achieve feature resolutions in the sub to tens of nanometer length scales without much difficulty. In terms of staining, you don't have to stain plunge frozen samples, though if your sample is fairly low contrast and won't really pop against the ice, we will generally, if you need your sample to be stained, so you need your sample to pop a little more against the background ice phase, you can do negative staining of plunge frozen samples to get that little bit of extra contrast. The main downside of plunge freezing is that it is bad for shear sensitive samples. The technique is pretty inherently shearing, so if your sample is going to do something weird or radically change its morphology when you shear it, plunge freezing may not be for your sample. On the other end of the spectrum, chemical fixation resin embedding can really accept any sample. So instead of just aqueous samples, you can also work with organic samples or dehydrated samples, as long as your thinnest length or your thinnest dimension is less than about a millimeter. In terms of the feature resolution, it's difficult to get features resolved that are less than about 10 nanometers in length scale. It's possible, it's just difficult. The main issues with chemical fixation and resin embedding is that you are going to have to stain your sample in order for it to show up against the resin background that you're embedding it in, unless your sample has some inherent contrast, for example, platinum crosslinks in a hydrogel or something. And due to the number of solution changes and processes that are done during chemical fixation, you do have to worry about many potential artifacts being introduced. These come from osmotic effects, washing out small molecules if you're looking for a particular small molecule in your sample. You can wash out lipids during some of the washing steps. Some of the stains we use have the potential to change protein conformations or depolymerize them entirely. And pretty uniformly, you're going to have some shrinkage or collapse of tissues, especially when working with hydrogels when you're doing chemical fixation. These aren't make or break things. There are ways of modifying the protocols and doing your chemical fixation in a way to kind of minimize these effects, but they're always something that is potentially going to happen that you have to worry about. And so if your sample is very prone to these, high pressure freezing freeze substitution is probably what you'll need to use. Like plunge freezing, high pressure freezing is limited to aqueous or hydrated samples. Unlike plunge freezing, you can really work with almost any concentration of sample, and higher is better. So if you have something that is 50, 60, 70 weight percent sample, and the remainder is water, that would probably be almost perfect for high pressure freezing. Additionally, because you do almost all of your chemical fixation steps at cryogenic temperatures in a non-aqueous medium, you avoid a lot of these osmotic effects, and so you can avoid a lot of the artifacts that chemical fixation can introduce. The downsides of high pressure freezing is that you are limited to relatively thin samples. Your sample has to be less than about 200 microns thick, but you can resolve features that are in the sub 1 nanometer length scale if you want to do high pressure freezing, fib lift out TEM. In those cases, you wouldn't need to stain your sample, though that is the most involved form of high pressure freezing characterization you can think of. Probably warrants an entire tech talk all on its own. If you want to do just a cross section, a single cross section, you can do high pressure freezing freeze fracture. For SEM, for those kinds of techniques, you don't have to do any staining. If you want to do a full Z reconstruction and look at multiple layers of your sample, you will need to do high pressure freezing freeze substitution, which will involve staining your sample. And we'll talk about that in particular because that is one of the things we do most commonly with high pressure freezing. Finally, it is worth noting for this introductory sample prep bit that if your sample is a little bit too large for high pressure freezing or chemical fixation, we do have a vibrating blade microtome that will allow us to cut soft matter samples down to the appropriate length scale. So if you're watching this and going, my sample is 5 millimeters in size, I won't be able to do this. We have a knife, we can make it smaller. It'll work, it'll be great. So into the actual protocols of how these work, let's start with plunge freezing. So when you do plunge freezing of a sample, you start off by pipetting about 4 microliters of your sample directly onto a TEM grid, then blotting that to thin your liquid layer down to about a couple hundred nanometers in thickness. You then will plunge your grid directly into liquid ethane. This freezes your sample so fast that there's not time for crystalline ice to form, and you instead have solely amorphous vitreous ice and your sample perfectly preserved inside of it. You then transfer your grid into a cryo-TM holder. It's not quite as clean of a transfer as this. You actually have to do 2 or 3 transfers to get it there because it's in a separate room almost all of the time. But then you can image your sample in a TEM under cryogenic conditions and get your features of interest very easily. So plunge freezing has a lot of advantages in turnaround time of sample and resolution. So a plunge freezing workflow is going to be on the order of a couple of days, whereas the other techniques are going to take quite a bit longer. The downsides are that plunge freezing cryo-TM is a technique that you really have to practice all of the soft skills a lot. These particular grid transfer changes in and out of the cryogenic phases take a lot of practice. So we are very happy to train you in how to do this. We're also very happy to do it for you. And if you want more details about this, we have a prior tech talk here at this QR code that you can watch and really learn more about that. Moving on to chemical fixation, as that really sets up high pressure freezing's complicated steps very nicely. Here we can really work with much larger samples. The problem is the artifacts. And this really comes from all the various steps we have to put our samples through when we are working through chemical fixation. So with chemical fixation, unsurprisingly, we start first and foremost with fixing your sample. So we're going to start with glutaraldehyde and paraformaldehyde. You want to use both of these fixatives to get your proteins to be well cross-linked and well fixed without being too hard or brittle. If you use only glutaraldehyde or only paraformaldehyde, you tend to not get very good ultrastructure preservation, so you do want to use a combination of both. It's also really important that you do this chemical fixation in a buffer. If you do it just in neat DI water, you are going to introduce a lot of osmotic effects, and your samples are going to be distorted in a variety of ways that are going to be very hard to tease out and understand after the fact. Once your sample's been fixed, we will then do a post fixation. So in the first chemical fixation, we're really focused on fixing proteins. In this post fixation, we're aiming it to fix all of your lipid bilayers. So osmium tetroxide is a very specific stain for unsaturated double bonds, which are principally only in your lipids. So we can get both some lipid membrane staining and labeling, as well as really fix those lipid bilayers and make sure they don't diffuse or get washed out later. We'll then follow this up with an N-block stain using uranyl acetate. This will give us really good Z contrast for our electron microscopy imaging. It also helps to stabilize a variety of negatively charged objects in the cell. And following that, we'll dehydrate our sample from aqueous medium into organic phase, either with ethanol or acetone, and then do a resin infiltration. This dehydration and resin infiltration step is going to be slightly different if you are starting with your sample in aqueous phase versus organic phase versus dried. If you're starting with your sample in organic phase, you can really skip this dehydration step and just go straight to resin infiltration. The other bit of nuance is that for dehydration and resin infiltration, you want to do as long of washes as possible to really remove all of the water or fully infiltrate the resin. However, you don't want to go too long because you can start washing out lipids or small molecules or other features in your sample. So it is better to have smaller samples, as we can then do slightly shorter washes. The larger your sample is, or the less permeable your sample is, the longer the washes will have to do, and that can potentially introduce some other artifacts. So this is where it really is helpful to use our vibrating blade microtome to get your sample as small as possible. Once your sample is fully resin infiltrated, we'll polymerize it at 65 degrees Celsius for 2 days, and then you'll have your sample chit embedded in resin, which you can then section with ultramicrotomy to do TEM or SEM imaging. Now, it's worth noting that if you don't get quite enough electron microscopy stain with the post-fixation and N-block staining, you can post-stain your sections with uranyl acetate and lead citrate for some additional contrast. The other thing that's worth noting is that each of these metal stains works slightly differently. So I am going to go into a little bit of detail how each of these works, and talk about some of their safety concerns because they are chemically pretty nasty. So we're going to start off with osmium tetroxide. It's usually the first one of these metal stains we use. It also has the most utility in also being a fixative. So osmium tetroxide, probably most of you are familiar with it from using the osmium coater. In solid phase, it's this osmium 8 tetroxide species. In water, it's this osmium 8 octahedral species. In particular, we're interested in this osmium 8 osmium 8 tetroxide species because that will react very selectively with double bonds in our lipid bilayers. This will then form an osmium 5-membered ring, which can then be hydrolyzed by water to produce a cis-diol. So if you are a chemistry background, this is probably what you're most familiar with osmium tetroxide doing. That then spits out an osmium 6, which can self-react to form a dimer, which then self-reacts to reproduce an osmium 8, which can then go back through the cycle, as well as producing an osmium 4 tetroxide. This osmium 4 tetroxide then will degrade into an osmium 4 dioxide, which is lipophilic. That will then partition into your lipid membranes and will aggregate there and give you your electron microscopy contrast and help pull these or hold these membranes together. If you need a little bit of extra osmium staining, you can use two different methods. The first is to use potassium ferrocyanide to kind of skip this whole lipid reaction step and just generate your osmium 6, which can then react as normally. The other option is to do what is called a OTO protocol, where you'll do a standard osmium fixing step. Then you'll use thiocarbohydrazide to essentially label all of the surfaces of these osmium aggregates. And then you'll do another osmium staining step to essentially relabel all of your osmium that has been functionalized with thiocarbohydrazide and really just get very, very heavy osmium staining. The downsides of osmium staining are really entirely around safety. Osmium tetroxide is very volatile and it is extremely toxic. So when you're using it, it's really important that you use it exclusively in a well-functioning fume hood while wearing full PPE. This is why if you use the osmium coater in EPIC SPF, that osmium coater lives in a hood, and we very, very strongly request that you never try and change the osmium crystals yourself. It's pretty nasty. It's also really important that you collect all osmium tetroxide waste when you're doing these processes in sealed containers, as it will evaporate and get everywhere. In terms of being mindful of safety, the first sign you really have that you've been exposed to osmium tetroxide is that your cornea will turn permanently black. This is very bad, so you really want to be very careful when working with this stuff. For uranyl acetate, it is much easier. It's also higher Z element, so you can get a lot more electron microscopy contrast. That said, it's not nearly as specific as a stain. It's much more prolific. So uranyl acetate will bind essentially anything that has a negative charge in your sample, whether those are proteins, lipids, glycoproteins, or nucleic acids. The downside of uranyl acetate is that it's not super soluble. So if you add too much uranyl acetate, you run the risk of getting these uranyl acetate needle structures, which can kind of ruin your characterization. That said, you can post-stain with uranyl acetate after you have generated your thin sections to get a little bit more uranyl acetate contrast. So that is a very common thing we do in order to get a little bit of extra uranium contrast into our samples. The safety issues with uranyl acetate are that it is slightly radioactive. It is uranium salt. Mostly, though, it's severely nephrotoxic. So you have to be very careful in handling it, to always be wearing gloves. And you have to be sure that, because it is slightly radioactive, you collect all uranyl acetate waste as radioactive waste. If you are doing these workflows in the BioCryo space, we do have waste streams for both osmium tetroxide and uranyl acetate. So just put it in the appropriate jar. And when you're storing uranyl acetate, it's important to keep it wrapped in aluminum foil as it will degrade when exposed to UV light. It's also slightly radioactive, so the aluminum foil is a safety thing for you as well, but mostly it keeps the uranium a little bit happier, so we can use it for a little bit longer. Finally, for the last post-staining chemical we will tend to use is lead citrate. It is similarly prolific to uranyl acetate, though it stains slightly different things. So ribosomes, lipid membranes, cytoskeleton components, and a variety of cytoplasmic compartments. It also will bind to reduced osmium as well as already bound uranyl acetate. So that's why we tend to use it last. It is, however, very finicky compared to the other two stains. So it's pretty water insoluble, and if you exhale on it, the carbon dioxide in your breath will cause it to precipitate. So you do have to be fairly careful in handling it. It's also extremely toxic, so you have to wear gloves and all of the various other things that you have to do with all these other metal stains we're using. When you use lead citrate, you tend to get one of two structures. You either get heavily stained, larger particles, or you get a very thin layer of what almost looks like looking down through smog over your entire section. So you can see areas where there's lovely lots of lead citrate staining, and then little white spaces where there's less lead citrate. In both these cases, you can actually get a fair amount of information just over how your sample is being stained to really get very nice images and learn a fair amount about your sample. So that's chemical fixation. Um, I just want to reiterate that a lot of these issues we have to do with not matching your buffers, or the potential issues you have with chemical fixation have to come from not matching your buffers properly and overwashing your samples. Or they're just unavoidable with chemical fixation because we are going to have to change phases if you're starting with an aqueous sample. To avoid a lot of those issues, you can work on refining your protocol a little bit. So if you are getting excessive metal staining, or you're getting a little bit too much of washing out of lipids or a small molecule you want to preserve, we can work on shorter washes and having smaller samples to kind of make sure that you're still getting proper infiltration. But probably what's going to be necessary is high pressure freezing freeze substitution. So high pressure freezing really leverages both the strengths of plunge freezing and of chemical fixation for larger samples. And this is because high pressure freezing takes advantage of water's particular weirdness. So water, unlike almost everything else, expands when it solidifies, whereas everything else usually contracts when it solidifies. This means that when you apply very high pressure to an aqueous sample during or prior to freezing, you actually suppress the freezing point, and it allows you to get vitreously frozen samples that are much thicker than otherwise would be possible. We do this using a high pressure freezer. The way this works is functionally you have two jets of liquid nitrogen that hit both sides of your sample, and that both increases the pressure substantially and cools your sample very quickly so that you can get very, very high cooling rates. So in the order of tens of thousands of degrees per second, while keeping your sample at very high pressure so that you have this prevention of ice crystals from forming. You then are able to begin your chemical fixation processes in high pressure freezing freeze substitution at those cryogenic temperatures. So unlike chemical fixation, where all of our chemical fixing is happening at room temperature, where diffusion is very prolific and you actually are working against these diffusion gradients, in chemical fixation, you'll etch out all of the water and introduce at the same time all of your chemical fixatives so that when you get up to a temperature where they are active and your sample is able to diffuse, your chemical fixatives will fix your sample long before they're able to go anywhere and any osmotic effects are able to happen. This works quite well. So to walk through this with like a particular sample, this is one of the spider silk glands that I worked with in my PhD here at BioCryo. So this is a spider silk gland that's been dissected out of a black widow. We then put it into a high pressure freezing planchette and fill the negative space here with a matrix. In this case, it's an agarose gel that we also will use BSA solution pretty often. Just something that will be somewhat ice crystal formation resistant without being too complicated or being something you'd mistake for your sample. We then use the science cannon. So our high pressure freezer is quite loud. So if you are in Hogan 5150 when it is working, it is normal to jump at least a little bit. But it does produce samples that are up to 200, maybe even 400 microns thick that are nicely vitreously frozen. We can then begin the freeze substitution process. So this is a uranyl acetate and glutaraldehyde solution in acetone, which we freeze in liquid nitrogen to form a slurry. You then put your little sample puck on top of that slurry and put it into the automated freeze substitution instrument. From there, it will slowly warm the sample up and etch out all of the water with the acetone, while also introducing all of your fixatives, which will then react once they get to their reaction temperatures. And you'll have a partial or well-fixed, partially stained sample in acetone at the end of that. You can then do your osmium staining. So in this case, this is osmium with a saturated ferrocyanide solution. So we can get lots of osmium staining to happen with this particular sample. So that's what this slurry of ferrocyanide is here at the bottom. The sample is somewhere in that. And then resin infiltration and polymerization as with chemical fixation to get your final sample chit that you can then section and get your TEM sections for either TEM or SEM and do your volume reconstructions. It's worth noting that there is some nuance in how exactly you want to do your end block staining after you finish freeze substitution. So if you want to make things a little bit easier for yourself, you can leave everything in organic phase. You tend to get not quite as much metal loading because those metal stains aren't quite as soluble, but you can still get very good contrast and staining. If you need very heavy staining, you can switch to aqueous phase. You can also switch to aqueous phase for your end block staining if you want to do some specific labeling. So to really highlight how this works, we can directly compare the two. So with high pressure freezing, once you've frozen your sample, you can store it on liquid nitrogen for days or weeks until you're ready to begin freeze substitution. At that point, you will etch out all of your aqueous phase and replace it with your organic phase and start your staining process. From there, it really depends on what specifically you want to do. If you just want to get a decent amount of metal loading and get your sample ready to go as quick as possible, you can do all organic phase and block staining and go straight to resin infiltration. If you want to do a little bit more heavily end block staining, or if you want to add specific antibody labels or do antibody labeling and then fluorescence microscopy, you're going to have to switch back to aqueous phase prior to doing your dehydration. Regardless, though, you'll end up with your heavily stained sample at about room temperature. And then, depending on whether you move back to aqueous or stayed in organic phase, you'll go back into the dehydration and resin infiltration workflow from chemical fixation and generate your final sections. So just to quickly summarize, and help you kind of pick which technique is right for you and your sample. Plunge freezing is going to work really well for dilute, aqueous, and very thin samples. If your sample isn't fairly dilute and in aqueous phase and very thin, it's probably not going to work at all. Additionally, it's not going to work very well if your sample is highly sensitive to shear. It does have the advantage of the highest resolution possible of any of these three techniques, and it keeps your sample in its native state the entire time, so you have usually the lowest chance of introducing any artifacts. Finally, it has the advantage of having the very fastest sample turnaround of any of these three techniques. Your typical plunge freezing cryo TM session is going to be on the order of 1 to 2 days for all told sample prep and imaging, whereas for high pressure freezing, freeze substitution, chemical fixation, you're looking at more like weeks. For high pressure freezing freeze sub, you can look at almost any aqueous or hydrated sample. You just need enough water content that we can freeze it. And you're going to avoid most of the artifacts that chemical fixation will introduce. On the downside, you're going to be limited to samples that are less than about 200 microns thick, and it is going to take a long time to turn around your sample. So a high pressure freezing freeze sub run is going to run more on the order of 2 weeks just to do your freezing, freeze substitution, and then your end block staining and resin embedding, which, as it usually takes multiple runs to get a protocol perfectly refined, means that these take a little bit longer. The other downside of high pressure freezing freeze substitution are these extremely toxic metal stains. You do need to handle them pretty carefully and be very mindful when using them because they are quite toxic and quite hazardous. Finally, chemical fixation and resin embedding, you can look at the largest samples and you can look at the largest diversity of samples. So aqueous samples, hydrated samples, organic samples, dried samples, all of these can really go through a chemical fixation workflow. It has the advantage of being about a 1 week turnaround for chemical fixation and resin embedding. And most of that time is the sample slowly being infiltrated with your fixative. So it is really only about 1 day of work and then several 1-hour sessions over the course of 4 days to kind of finish up the process. The downsides are that it has many the same highly toxic metal stains as high pressure freezing freeze substitution, and there are a lot of potential artifacts that can happen during chemical fixation and resin embedding, be they osmotic effects, washing out small molecules or your lipids, conformational changes in proteins, or entirely protein depolymerization, and shrinking and collapse of tissues and hydrogels is very common. So it's worth trying before you do high pressure freezing freeze substitution, if only get an idea of how your protocol works and what artifacts or staining it provides. But it may cause problems that will force you to use high pressure freezing freeze substitution. Regardless of which technique you think might be best, we're very happy to work with you on any of them and to really help you pick and design and test a protocol for your sample. It is very much more art than science for a lot of these, where you will look at a particular protocol that has been shown to work for a particular sample that is similar to yours, and you'll try it and it won't work, and then you'll try a couple other things and find something else that works quite well. We have the advantage of having done this on a variety of samples, so we have a little more intuition on what um, things have worked for us before. That said, if you have a protocol you want to try, please do bring it by, and we're happy to work with you to implement it. So with that, if you have any questions, please don't hesitate to reach out to us. BioCryo as a whole is happy to schedule a meeting to discuss your samples or any technical questions you have. This QR code will take you to our BioCryo webpage, and if you want to email us, my email is here, as well as Reiner's and Eric's. And then on that note, if you have any other questions that you want to ask now, please feel free. Or if you just want to email them to me, that is also fine. And if you want to look at any of the other tech talks that we have, they all live at our Nuance YouTube page here. And you can learn a lot more about the other instruments and techniques we have here at Nuance. With that, thank you.